Hello, I'm Pattrick Burke. Today is August 12th, and this is the video lab exercise for the digestive system, week 6. So we're eating a cheeseburger, and we break off a piece of the cheeseburger into a size that we can masticate, and we then our salivary glands and lingual lipase has already been started to be produced when we saw the cheeseburger, or even when we thought about it, and that's mixing with the polysaccharides in the bun and to form disaccharides and trisaccharides. The teeth are masticating it up the bun and the tomato and the meat and the cheese and onion and maybe mayonnaise and whatever for the more lipids in there, so then the tongue is going to form a bolus. We're going to masticate. We're going to then swallow that through deglutition into the esophagus, and the peristalsis is going to carry that down to the stomach, where it's going to encounter the mechanical churning. It's going to mix with gastric lipase there, and we're going to start to break down the fats then, and into the triglycerides and fatty acids and monoacylglycerides. The meat is going to be, gastrin is going to be stimulating hydrochloric acid production, and that is going to activate the pepsin, which is going to break down those proteins into peptides, and the bicarbonate is going to be buffering that process so it doesn't become too acidic for the stomach lining. And then it's going to, once it's kind, it's going to be moved into the small intestine, the duodenum, ileum, and there it's going to encounter the products of the pancreatic actinol cells. That's going to encounter the pancreatic amylase, which is going to break down alpha dextrinase, is going to break down the alpha dextrins into glucose. The enteropeptidase is going to break down trypsinogen and then into trypsin. The cheese, you ate cheese on that burger, so we've got lactase necessary to break down the lactose into glucose and galactose. And the nucleosidases and nucleotides are going to break down into nitrogenous bases and pentoses. And the peptidases are going to break down into the amino acids. Also, that pancreatic lipase is going to mix with where now the bile salts have emulsified these triglycerides and form them. There it forms into emulsify the triglycerides into the fatty acids into monoacylglycerides. Monoacylglycerol, and forms these micelles for transport through the epithelial cells in the villi, where they are incorporated as triglycerides mixed with phospholipids and cholesterol in the Golgi apparatus, and give it a protein coat. Now this is called a chylomicron, which is ready to pass into the lacteals into the lymphatic system, and then from there it will be going into the thoracic duct to return into the bloodstream. Meanwhile, and that's going to be via once it's in that state, it can absorb by passive diffusion, because it's going to be going down the concentration gradient. Now, a lot of things are going to be going up the concentration gradient by active transport. Nucleic acids, for instance, and fructose is going to be going in by using a carrier protein by facilitated diffusion. Carbohydrates, glucose, galactose, and proteins are going to be mostly going in by co-transport using passively diffusing molecules as carriers, such as sodium. And then some things are going in by endocytosis, in which the cells actually engulf the nutrients, so that the non-lipid nutrients are then absorbed into the capillary blood, which will then enter the hepatic portal vein, and then they get to where this is all kind of heading, which is into the various processes of the Krebs cycle. Once they're broken down into small enough molecules, glucose can be used. Glucose, amino acids, and glycerols and fatty acids can all be used for energy production. The lipids can be used in the to make acetyl coenzyme A when the glucose is limited, and that can then either break down adenine triphosphate to form energy, or the amino acids can be used for protein synthesis, or the lipids can be synthesized and stored. That's how that works. Thanks.